Discovery and Development Program.
Sound okay?
Yes po.
Okay. So, good morning, everyone. So, I'll be your resource speaker for the entire training today. So, I'll be discussing on two topics. And of course, I think drop the formalities. You can just call me Amy. And let me share with you my experience on lab animal research. And I've been conducting animal research, I think, for the past 30 years. And of course, both for my graduate degree research as well as when I came back after my PhD abroad. And I'll be sharing with you all the experiences and how we are handling, of course, our animals, and starting off with laboratory animals in research. So let me share my screen to start my first talk. Okay. Okay. Now, my first talk is all about laboratory animals in research. So this would be the topics that I'll be discussing. So we'll start with the importance of animal in research, also the proper animal modeling, and we'll delve on the specific laboratory animals and the different animal models that can be used for specific research and testing. I'll be touching on the specific strains or breeds for larger animals. What are the specific characteristics or phenotypes of these laboratory animals or models? And what are the specific uses in research? I think I've concentrated on specific researches that are being conducted by the Tuklas Lunas centers in the Philippines. So it's a combination of metabolic diseases, cardiovascular diseases. We'll touch on cancer, and of course, the safety evaluation testing that are needed under FDA approval. Now, I think the basic question is why are we using animals in research? So the reason for this is because the diseases or the systems that we would like to know is actually very much similar with humans. So the homology in terms of the biological system between animals and humans is actually 98%. And you just need to choose the right animal model for the specific disease that you want to do research on. In terms of susceptibility to diseases, so animals are comparable in humans. And this holds true whether these are metabolic diseases such as diabetes mellitus, cardiovascular diseases such as hypertension, stroke, dyslipidemia, cancer, infectious diseases. Now, the reason why we're using animals in research is because in spite of development of in vitro systems, whether these are cell-based, tissue-based, and right now, the organoid system, so these cannot substitute to animal experimentation. So we can do collect very important data, but still, this would require animal testing. Now, there was also development of in silico models, or you use models using computers. No? I think this is also very helpful in research, but again, there are certain limitations for this computer modeling. So which means that we really need to require in vivo or animal experimentation. And of course, when we conduct such animal experimentation, I think this was emphasized by Dr. Saloma, that we need to follow the 3 R's principle of Russell and Birch. So just to give you an idea, way back in the 60s up until the 1990s, so in order to properly conduct animal experimentation, and get validated data, you need to use a great number of animals. So you'll be using hundreds or even thousands of laboratory animals. But right now, because of the strict implementation of the animal welfare law globally, so we need to reduce the use of laboratory animals, but still get very important data for our research. Now, specifically, we use animals in biomedical research. And the reason for this is because a lot of the researches on biomedical science, we cannot go directly using humans. So it's more ethical in order to use animals for our experimentation and collect viable information before we proceed with the clinical trial in humans. So that's the reason why preclinical studies using animals are very important for biomedical research. Now, of course, there's a drawback in using animals. No? So we would like to hope that there is a single ideal animal model for a specific human disease. But unfortunately, there is no single animal model that we can use. And that's the reason why there are a variety of animal models that we have to use in our quest for biomedical research. So the reason for this is because no single animal model can replicate the human disease in terms of its pathophysiology, its ethiogenesis, the biomarkers, the toxicity, and other complexities of the human disease. So that's the reason why it's always a step or a process when you conduct a biomedical research. Now, animal biomedical research can be divided into basic and translational. Uh, and I think in the Philippines, we're doing a combination of both. Okay. So in basic research, we do conduct this using animals to further understand the physiology, the pathology, and the biological system interactions in a specific disease condition. And in translational research, in using animals, so this is where we conduct our studies in order to demonstrate the efficacy, to document any side effects or toxic effects of possible new compounds, drugs, or therapies for a specific disease condition. So it's not only confined to drug development research, but biomedical research is also targeted on developing diagnostic procedures or even test kits, surgical procedures that is addressing a specific disease condition. Now, this is quite an interesting paper that I think any person that is into biomedical research, especially drug development research, should read on. This is a review article on the correlation between animal modeling and drug efficacy results. So this is a retrospective study that was done on the 100 best-selling drugs for humans, and it is addressing specific disease condition. And based on this review article, so they were able to see the positive correlation between using the right animal model and the high drug efficacy that was um, uh, stated in this review article. So based on this, the specific take-home messages for this review article is that good drug efficacy for specific diseases is dependent on using the right animal model to test it. It was also able to reduce the cost of drug development research, and at the same time, based on the important findings, it was able to reduce the risk of human subjects that were included in the clinical trial studies. So which means that you have to choose the right animal model for your biomedical research. And what you can see here are the specific laboratory animals that are being used in biomedical research. So we can classify them as small laboratory animals. So specifically, you have the rodents as the most popular and most commonly used. So we have, of course, the mice and, of course, the rats. We also have the hamsters and, of course, the guinea pig and, of course, the rabbit. And, of course, the second tier for biomedical research would require the use of larger animals. And the dog, specifically the beagle, is a very good animal model for specific biomedical research. The mini pig is also specific for certain biomedical research that we would be discussing. And of course, the non-human primates is also popularly used, especially for the COVID vaccine development that was done by a lot of pharmaceutical companies globally. Now, just to give you an idea, this is a 2000 data in terms of the global usage of laboratory animals. And as you can see in the pie chart, so global usage around 95 to 96% would be the rodents. So the reason for this is because animal research is very expensive. And among the different laboratory animals, the mice and the rats are the least expensive compared to the other laboratory animals. And that's the reason why initial biomedical research uses the rodents. Now, there is an increasing trend in the use of large animals, such as the dog, the pigs, and the non-human primates. Because based on the pathophysiology of human diseases, there are specific diseases that mimics the human diseases in using these larger forms of animals. Now, with regards to the hamsters, the rabbit, and the guinea pig, there are specific biomedical concerns or researches that is appropriate to use these animals. And again, we would be discussing this in this presentation. So let me start off with one of my favorite animal to use in biomedical research, and I think globally this is the most commonly used. That would be the laboratory mice. And the reason why it's popularly used is because in terms of anatomy, physiology, and genetics, they closely resemble that of humans. And remember that the genomics of the mouse and the humans were completely studied way back in the late 1990s. And based on those studies, 95% of the 30,000 genes are shared by both the mice and humans. And this has been published by a lot of scientists in the journal Nature. Now, the reason why laboratory mice are very popular and very advantageous to use is because in terms of the resources to maintain them, so this is quite minimal or of a less cost compared to the other animals. So in terms of your operating expense and to maintain a colony or a group of animals, it's easier. Another thing, it's easy to reproduce the animals because physiologically, they have a shorter pregnancy and nursing period. So which means that in terms of a breeding colony, you would be able to produce more animals for a shorter period of time. And they also have a high number of offspring depending on the strain of the mice. So which means, again, very advantageous in terms of providing the adequate number of animals for your research endeavor. They also have a shorter life cycle. So which means that if you're doing, say, reproductive studies and you need to collect embryos or follow through from birth to adult or even old age of the animal, it's very convenient to use. And another thing, they are now being used for the past 30 years in the development of what we call knockout or transgenic models of human diseases. Now, right now, there are around four groups of laboratory mice that are available for our biomedical. With a colony of our transgenic or knockout strains. Now, the good thing about this, so the animal geneticists globally are actually selling or using the commercial animal vendors globally in order to preserve their transgenic or knockout strains. So all you have to do is order the particular transgenic or knockout strains of mice that you want to use for your particular research. And of course, these congenic strains, okay, so this is what we call a repeated backcross, wherein we do the breeding. And as I've said, the most popular to use would be the C57BL6 or popularly called the B6. And this requires about 10 generations in order to produce the congenic strains. Now, just to give you an idea, this is the data that I got from PubMed. And this is the types of mice and the frequency of citations in the PubMed website. This is starting from year 2000, year 2010, and the latest data is for the year 2018. And if you look at the types of mice, so from inbred, transgenic, knockout, congenic, or mice in general, so there's an increasing trend in terms of the publication and the frequency of citation. So which means that the global usage of laboratory mice for research, specifically biomedical research, have been increasing tremendously for the past decades. And if you look at the data, there's an increasing trend, especially on the use of transgenic or knockout or inbred strains from these particular decades. Now, starting off with the outbred strain, I think this is the popular outbred strain that we have in the Philippines, and this is what we call ICR. So this particular strain was developed or originated in Switzerland, but it was in the Institute of Cancer Research in the United States, wherein they were able to particularly breed and make a colony of these ICR mice. So if you're buying your mice from the RITM in Alabang, this is the particular albino mice that you are actually purchasing. So they are very easy to handle. They have a very good temperament. They're very docile, so non-aggressive, and they have a very low incidence of spontaneous tumors. So in terms of their usage, so ICR mice are commonly used for toxicology research. If you're doing all those safety tests that are required by the FDA, such as the oral toxicity testing, whether it's acute, subchronic, or chronic, and systemic toxicity testing, then the ICR mice is a good choice. It can also be used in certain pharmacological research, but this is limited in neurobiology research, epidemiology, and it can even induce infectious diseases in ICR mice. We've done a lot of researches and testing for the industry, specifically for FDA safety evaluation test. Now, one of the popular inbred strain would be the C57BL6. So it's popularly called also C57 Black 6 or B6. So the reason why it's called B6 is because of the black hair coat of this animal. So it was developed by Dr. Little in Harvard University, and the same fellow is the founder of one of the biggest global animal provider in the world, and that is the Jackson Laboratories, where we can order our laboratory animals. So there are different substrains of the B6. So usually for biomedical research, we use the wild type as the control, but you can also buy congenic and transgenic strains. So again, it has a very good temperament. It's very stable, and it's very easy to breed. And this is popularly used for a lot of biomedical researches. So we've done researches on pharmacology, also physiology. This is also a good model for type 2 diabetes mellitus. This is also popularly used for eye problems such as glaucoma, retinal problems. It's also a very good model for cardiovascular problems such as atherosclerosis or diet-induced obesity. So if you want to create an obese model using a high-fat diet, the B6 is a good model to use. And this is popularly used also for Alzheimer's and Parkinson researches. Now, another popular inbred strain would be the Bob C. So in the Philippines, RITM is producing it. Unfortunately, they are not selling it, and they're using it for their in-house researches. And the reason for this is because the Bob C is a very popular strain for immunology research. So this is specifically used for monoclonal antibody production as well as hyperserum. This is also good to use in hybridoma researches. It's also a very stable strain for tumor or cancer research. So if you're going to do transplantation of tumors or injection of tumor cells, This is also a very good model to use. And this is also a stable strain if you want to do infectious diseases. So they also have a very good temperament. So it's very easy to handle, non-aggressive. And of course, the reason why tumors, no, or hybridomas grow on them is because they are immunodeficient. And it was developed, of course, by the National Institutes of Health in the United States. Now, I think a lot of the Tuklas Luna centers are using the diabetic models for research. And I think you need to know what would be the best model for the particular research that you are using. Now, you have a choice of inducing it chemically, and I know that a lot of the Filipino scientists are doing this with the use of aloxan or streptozotocin or STZ. Now, remember that this chemically or pharmacologically, or you call it iatrogenically induced diabetic model, is a replicate of type 1 diabetes mellitus because you are practically destroying a larger population of the pancreatic beta cells when you give aloxan or STZ. It's a very simple model of hyperglycemia, and this is the model that is usually used if there are new formulations of insulin that are coming out in the market. So they use this for testing. But remember, if you're thinking of researching compounds or extracts or drugs that would have a different mechanism of action, then this is not the model to use, especially if you're trying to check on insulin response, because you are practically destroying the pancreatic beta cells with these pharmacological drugs. Now, another group of model that you can use for type 1 diabetes mellitus would be a polygenic spontaneous autoimmune type 1 diabetic model. And this is what we call the NOD mouse. And you also have a choice of the BB rat. So specifically, that's why it's called autoimmune model. So the body of the animal is eliciting an autoimmune response to the pancreatic beta cells. So it's recognizing the pancreatic islets as foreign. So it's eliciting a leukocytic infiltration to destroy it. That's why we call it as a model of insulinitis. So ITIS or itis is what we call inflammation. It's a general term that we use for inflammation. So we're literally creating inflammation specifically of the pancreatic islet where you can find the pancreatic beta cells. So this causes high blood glucose level or BGL and low blood insulin levels in this particular NOD mouse and BB rats. And in biomedical research, this is being used for the studies of the genetics and the pathophysiology of type 1 diabetes mellitus, because a lot of the type 1 diabetes mellitus is because of genetic mutations, whether these are genetic mutations in the insulin gene, it's a mutation in the pancreatic beta cells, mutation in the glucose transporters, and also mutation in the insulin receptors in the target cells. And this is also used to evaluate or test drugs or new therapies for type 1 diabetes mellitus. Now, there is also genetically induced, this is the Akita mice, wherein it has a mutation in the insulin gene 2. So specifically, the mutation causes a defect in the proinsulin. So this is the immature protein that would give rise to the mature insulin hormone. And specifically, what they've seen are the misfolding of the proteins of the proinsulin. So the phenotype of this particular Akita mice would be the lack of pancreatic beta cells. So the animal would have a high BGL, insulinemia, so very low or non-detectable insulin levels, and we have the polyuria polydipsia syndrome. So increased urination and increased drinking of the animal. And this is the biomedical model to be used for transplantation studies, specifically of the pancreas. Model for the type 1 diabetic macrovascular disease. So the effect on the blood vessels, specifically atherosclerosis, and for diabetic neuropathy. So the neurological signs that we can see in patients that are afflicted with type 1 diabetes mellitus. Now, there is also a model for pre-diabetes, and these are the diet-induced obesity mouse model. And we do have several types of type 2 diabetic models. So they are classified as the monogenic and the polygenic diabetic models. Now, when we talk about the pre-diabetic mouse, no, we want this. Why? Because we want to catch the diabetes before it develops into a full-blown diabetes mellitus. And as I've said, the C57 BL6 or the B6 is the congenic strain that is being used for this particular modeling. So you give a high-fat diet. That's why it's called DOI, diabetic induced obesity. So there's a special diet that you can use for this B6 mice, and you would be able to develop this pre-diabetic model. And this is popularly used to develop or study the pathogenesis of pre-diabetes and also diabetic neuropathy. So usually the progression of the neurological problems from early or young animals until their adulthood. And since this is also diet-induced obesity, this is also popularly used as a model for the studying of obesity. Now let's talk about the monogenic type 2 diabetic mice. And one of the model has a mutation in the OB gene or leptin gene. So if you're familiar, leptin is a hormone that is produced by the fat tissues, both the white and the brown adipose tissues. But there is greater amounts of leptin that is produced by the white adipose tissues. So leptin is a hormone that is produced by this tissue when the animals are obese and even in humans. And they control the obesity by reducing the feed intake of the animal and increasing metabolism. So in this particular strain, there is a defect in the leptin or the OB gene. So that's why it's called OB-OB, as you can see in the photo. So this particular mouse strain is morbidly obese. And they are obese because they are hyperphagic. So their feed intake is uncontrollable. So that's why they're gaining the weight. And of course, they have a high BGL, high insulin levels. They develop insulin resistance. And of course, synonymous with diabetes. So when they get wounds, there is a delay in healing. You cannot breed them as well because they are sterile. So this is a very good model of phase 1, phase 2, and phase 3 type 2 diabetes mellitus. And this is also a very good model that is being used to test new compounds, drugs, or therapies for type 2 diabetes mellitus. You can also use this for obesity research. And if you are interested on wound healing studies, if you have a biomedical product for wound healing and you want to test it, since diabetes-induced wounds are long-term, then you can also use this particular model. Now, let's talk about another monogenic type 2 diabetic mice, and that would be the DB mice. So the mutation of this mouse is on the leptin receptor. So the leptin receptors, in terms of their target sites, in order to control feed intake, would be the neurons in the hypothalamus. So they have receptors and specific neurons in the hypothalamus, specifically the feeding center and the satiety center in the hypothalamus in the brain. So as you can see, because the leptin cannot bind to the leptin receptors, so they also develop hyperphagia. And with this increase or uncontrollable feed intake, so the mouse develops obesity. And there is a very high blood glucose level. Eventually, there would be pancreatic beta cell atrophy, and there would be low insulin levels. And similarly with the OB mouse, this is a very good model for phase 1, phase 2, and phase 3 type 2 diabetic mellitus. So this is popularly used to test drugs, compounds, and therapies for type 2 as well as for obesity. And this is also a very good model for wound healing studies using the diabetes associated or induced wounds. Another monogenic type 2 diabetic mice would be the KKAY mouse. It has developed insulin resistance, and they have an agouti gene mutation. So the mouse here is the one colored brown or light brown, as you can see. So these mouse do not develop obesity, but they do have a high blood glucose level and insulin level. So they develop glucose intolerance and insulin resistance. And the reason for this is because the mutation causes pancreatic beta cell degranulation. So it's a pathology causing non-functionality of the pancreatic beta cells. And the mouse also develops mild liver steatosis or fat accumulation. So this is also a very good model for phase 1, phase 2, and phase 3 type 2 diabetes mellitus. So that's why you can also use this to evaluate compounds, drugs, or new therapies for type 2 diabetes mellitus. Now, we do have polygenic type 2 diabetes mice. And if you consider the complexity of the diabetes in humans, so the polygenic type 2 diabetic mice are more advantageous to use compared with the monogenic. So one of the strain is this one. As you can see, it has a very long coding because of the mutations. But this particular model develops obesity, hyperphagia. So it has a very high BGL. It has an elevated insulin, leptin, and triglyceride levels. So this is popularly used as a very good model, again, from phase 1 to 3 type 2 diabetes mellitus, as well as testing new compounds, drugs, or therapies for type 2 diabetes mellitus and obesity. Now, the other polygenic type 2 diabetic mice is this one. This is the Taliho J&J model. It's among the polygenic type 2 diabetic mice, this mimics many of the clinical signs or characteristics of human type 2 diabetes mellitus. And this is the very or most popular polygenic type 2 diabetic mice being used globally. It's a very good model for phase 1 to 3 type 2 diabetes mellitus. And this is being used by pharmaceutical companies globally for evaluating new drugs and therapies for type 2 diabetes mellitus. So the complete clinical signs of hyperglycemia, hyperinsulinemia, hyperlipidemia, moderate obesity, and the pathology in the pancreatic islet would be enlargement, which actually compromises pancreatic beta cell function in this particular model. So with that, you now have several diabetic models that you can choose from. Now, one of the research that are being conducted by Tuklas Luna's centers in the Philippines would be cancer. And right now, the trend is using immunodeficient mice for orthotopic cancer models. When we say orthotopic, this is either transplantation of the tumor, injection in the origin of the cancer. And right now, there are four general cancer models to choose from. No? So for here in the Philippines, I think popularly used is the nude mice. But I think we should take advantage of the newer cancer models, specifically the SCID, the RAG, and the multigenic deficient mice models. So for the nude mice, well, that's why it's called. Now you can see the picture; they are hairless, and that is because of the mutation in the new gene or the FOXN1 gene. But more importantly, the mutation would be the absence of the thymus gland. So they are athymic. So if you remember your physiology, the thymus gland is the gland that produces majority of the T cells or the T lymphocytes that we need for cell-mediated immune response. So this particular mouse strain would have very low T cells. And the T cells are the ones that can be what? Classified as killer T cells that can kill the cancer cells. There are also what we call uh, helper T cells that can stimulate the B cells for antibody production. And there are also T cells that are can produce the lymphokines, such as interferons and other substances that can also aid in cancer response. Now, the problem with the nude mice is that when you do the transplantation or injection of the tumor cells, there is a variability in tumor growth. And this is only a good model when you engraft or you inject fast-growing tumor cells, whether these are primary tumor cells, human-derived or mouse-derived. The slow-growing tumor cells usually have variable growth in the nude mouse. And that was the reason they developed the other newer orthotopic cancer mouse models. So these immunodeficient mice would be the SCID. So SCID referring to severe combined immunodeficient mouse models. So it has a mutation in the PRKDC. So this is the SCID locus specifically in chromosome 16 of the mouse. And the good thing about this, they are immunodeficient and they lack both the T cells and the B cells. So they don't produce the T lymphocytes and the B lymphocytes. So which means that if you transplant or inject the tumor cells, whether these are slow growing or fast growing tumor cells, then it can grow in this particular model. Okay? And this is a very good model for leukemia and lymphoma studies, aside from the other cancers. No? However, this is radiation sensitive. So that's the reason why if you plan to use radiation as part of your protocol, then you have to take note that the skin is radiation sensitive. Now, do we have another model for this? So one would be the RAG. So there are two types. We have the RAG1 and the RAG2. And this particular immunodeficient mouse also lacks the T and the B lymphocytes. And this is radiation insensitive. So that's why you have the option, okay? It's also a very good model for both the fast and the slow growing cancers or tumors. It's also good for the studies of leukemia and lymphoma. Now, we also have what we call the multigenic immunodeficient orthotopic mouse model, and this is the NSG. No? So it actually has mutation. So it is a SCID model with additional interleukin 2 receptor mutation. So it means that characteristically this mouse would be severely immunodeficient because it's also not producing T and B cells, and it prevents cytokine signaling, particularly interleukin 2. So it means that in terms of characteristic, this mouse, they do not produce T and B lymphocytes as well as natural killer T cells. So it's a very good model for both fast and growing cancers. This is also being used for human stem cells and grafting, for blood mononuclear cells implantation, xenograft studies, whether these are skin, tissues, or organ grafting, as well as adult stem cells or tissue transplantation studies. So it's not only confined to cancer research, but it's very good for immunology, stem cell research, and xenograft studies. So this is one example of that. This is the CB17 SCID mouse. So depending on commercial animal vendors that you would use, so may coding sila for that, no? And this is a very good model that is being used for cancer research, specifically tumor transplantation, for the testing of new cancer drugs and therapies, as well as for human immune system tissues. Now, just to share with you is modeling good. So let me share with you our success story in using these different animal models. So we do have a cancer research project that we have been doing for the past 4 years. And this is in collaboration with the Laboratory of Animal Modeling at the University of Tokyo, with our primary collaborator, Dr. Shigeru Kakuta. And what we use here, so we're very much interested on gastric adenocarcinoma, stomach cancer. This is a metastatic type, and we're using the A4GNT knockout mouse model. This is a transgenic spontaneous model for gastric adenocarcinoma that was developed by our collaborator at the University of Tokyo. So it's not commercially available. They don't want to sell it to the commercial animal vendors. And for us to gain access, of course, to this particular stomach cancer model, so we did a collaboration on our studies on the Philippine stingless bee propolis. And this particular study was done by our PhD student, Dr. Mark Desamero. So this is a 2018 publication that we have, and we have a continuous collaboration in terms of this particular research. We also I did a lot of studies way back in 2000, early 2000 on diabetes, but I think there are a lot of Filipino scientists conducting diabetes. So I think this was the last one that we did. This is using the Aloxan model for type 1 diabetes mellitus, and this was done with my MS student, Dr. Tangkilot of the Pampanga Agricultural State University. And this was published in the Journal of Medicinal Plant Research. No? We are also doing studies on inflammation. So this is the first screening that we did for the Philippine stingless bee propolis. This is a very good model for screening inflammation. This is the carrageenan-induced hindpaw edema model, and we tested the cytokines that are involved in inflammation. And this is an animal model that we did. And again, we published this just last year, 2020, in the journal Experimental Animals. So let's move on to the laboratory rats. Okay, so this is the second commonly used laboratory animal for research. It's a very good model for cardiovascular diseases because the rats are bigger. You can collect more blood. So in terms of checking the parameters for obesity, this is more advantageous than using the mouse. No? We're also using it for our hypertension studies. This is also a very good model for metabolic diseases, including diabetes mellitus. We're also using the laboratory rats for our stroke or ischemia models together with the Department of Neuroscience of the Philippine General Hospital. We're also doing behavioral studies. Rats are very good models because they mimic the behavior of humans. And a lot of the neuroscience work are using laboratories rats because of their bigger size and resistance, of course, when you're doing brain or stereotaxic surgeries. So there are, again, laboratory rats are classified as outbred, inbred, or transgenic, and we will be discussing some of them. So starting off with the Sprague Dolly rats, no? So the Sprague Dolly was developed in the US, but this is derived from the Wistar rats from Japan. And if you're buying your rats before from FDA in Alabang, so this is the strain of the rats that they have. It has a very good temperament, very easy count to handle. They also have no incidence of spontaneous tumors, and they are being used in various types of researches such as pharmacology, as I've said, metabolic diseases, behavioral, endocrinology, physiology, and I've done a lot of work on neuroscience, especially brain surgeries with this particular model. Now, the Wistar rats is the first rat strain that was developed as an animal model, and this originated in Japan. But right now, of course, it's all over the world, and there are also substrains right now that were developed from the original strain in Japan. It's a very, very good model to use. Again, it's very easy to handle. It's very calm. It has a very high litter size, so madaming offsprings, and that's the reason why it's very easy to produce. And again, low incidence of spontaneous tumors. Aside from what I've mentioned in terms of physiology, behavior, pharmacology, and neurosurgery and neuroscience, we also use the Wistar rats for collecting brain cells for cultures. So the Goto Kakizaki rat or GK rat is a non-obese Wistar substrain, and this is a type 2 diabetic rat model. So they develop this particular clinical signs of nephropathy, so the kidney signs, retinal problems, and cardiac complications of type 2 diabetes mellitus. So if you want to use a bigger model for type 2 diabetes model with these clinical symptoms, this is the rat of choice. So you can do these researches on this complications of type 2 diabetes and evaluate drugs, compounds, or therapies for these particular complications. Next, we've been doing researches on hypertension, and this is the model that we're using right now. This is the spontaneously hypertensive rat that was developed in Kyoto University. This is spontaneously hypertensive. So usually their blood pressure is between 140 over 160 or 140 over 200, no? All throughout. So it's a very good model for genetic hypertension. And right now we're doing a lot of research on hypertensive drug compounds, no? And this is the popular model to use for drug research development for hypertension. So if you want a combination of hypertensive and heart failure rats, then this would be your model of choice. This is the spontaneously hypertensive heart failure rats that was developed at the University of Chicago. So this is a very good model that is being used for heart failure. Remember, it's always it's a combination of hypertension and heart failure. So that's why you can use this one for pathogenesis, and you can also use this one for drug testing. And by the way, these animals are also type 2 diabetic. So you can also use it if you want all those complications in a model. Next is if you want a spontaneously hypertensive obese rat, then this would be your choice that was developed from Case Western Reserve University. So this has the phenotype of being hypertensive and obese. and even developing kidney problems. And this is the model that is being used to test drugs for obesity and hypertension. And remember, they go side by side. Next would be the Zucker rats. So this was developed by Eli Lilly or Elanco. This is named Elanco now. It's a big pharma company in 1974, but it was the Indiana University that did develop the inbred strain for the Zucker rats. So ang cute nila, no? They have a black hood, and this is a hypertensive, obese, diabetic rat. So you can use this for researches on this particular diseases, especially for testing of drugs and therapies for also for type 2 diabetes mellitus and hyperlipidemia as well as hypertension. So just to show you again, sharing with you our success in the use of animal models. This is a collaboration that I did with the Philippine Nuclear Research Institute, the group of Ma'am Chat Aranilla and Dr. Lucille Abad. So they developed an hemostatic medical product in order to address bleeding. So we tested it. So I did the rat bleeding models for them, as well as, of course, the toxicity testing as required by FDA. And this was a project funded by PCHRDDOST, and this is our 2020 publication. It's the Journal of Biomaterials Application. We also did, as I mentioned, we're doing work on cerebral ischemia or stroke. So we have a rat ischemic stroke model. It's a surgical model that we developed. And this is a collaboration that we did with the group of Dr. Dan Jestro of the Department of Neuroscience of the Philippine General Hospital. And this is our 2020 publication. We also have recently, it's a March 2021 publication. This is our study on legumes. It's a review article that we did with on spontaneously hypertensive rats and even the genomics. This is together with a group of Dr. Torio of the Institute of Chemistry of UP-LB and the Philippine Genome Center Agriculture Group. And this is in the journal Applied Science. So it shows that there is a good success if you use the right model. Now, for those that would be working on immunology research, particularly development of polyclonal antibodies, vaccine development, eye diseases, ophthalmology, and for those topically applied substances, so dermal toxicity and dermal irritation, the laboratory rat would be your choice. And the albino model is the best choice. What you can see here is a picture of our New Zealand rabbits that we buy for we're doing dermal toxicity, dermal irritation testing for products for commercial companies. Now, the laboratory guinea pig, there are 13 strains, and what you can see on top is the Hartley. This is the most popular strain to use for biomedical research. So dermatology and skin problems, laser surgery development. So they use this model. And the skin sensitivity testing that is required by FDA. So the Hartley would be a choice. And the guinea pig is a very good model of asthma. It's also a very good model of deafness for noise pollution and for checking ototoxic drugs. No? It's also a very good model for wound healing as well as eye problems. So such as myopia, glaucoma, and so on. So what you can see is the IAF hairless guinea pig that is being used for dermatology and skin problem research. The laboratory hamster is being used for carcinogenicity, infectious diseases, but I think let me make mention that this was popularly used for SARS-CoV-2 research because they have a high sequence homology with the human ACE2 receptors. No? And when you inoculate the virus, they develop high viral titers within 48 hours after inoculation. And they also mimic the clinical symptoms or the pathology such as cytokine activation, spleen and lymphoid develop serum antibodies. So before going into non-human primate testing of the COVID vaccines, so they use these laboratory hamsters for COVID, SARS-CoV-2 research. Now, the Beagle was developed, first initially used at the University of Utah, and now it's a very good model in various biomedical research, most especially the bioequivalent studies. So because the bioequivalencies for the Beagle is quite similar with humans. The development of pacemakers that we have now for heart patients were also developed using the Beagle. Periodontal diseases and all those invasive surgeries, such as the coronary artery bypass that is being used for human patients, were initially developed using the Beagle. The mini pigs are very cute. What you can see here is a mini pig. So depending on the country, these are the countries that develop the various. So in the Asia Pacific region, so we have China with 3 models and the Lanyu mini pig from Taiwan. Okay. And for research purposes, they are very good for ocular and cardiovascular therapeutics. The pig is a very good model for myocardial infarction and the best model for diabetes. So usually po in diabetic research, you start with either the mouse or the rat, and before you do the clinical testing, you have to use the mini pig model also. So it's a very good model for glaucoma. It's a very good model for the various diagnostic techniques that we're using now in human medicine. Electroretinography, tomography, endoscopy, angiography, the laser therapy. So we use mini pigs. No? It's a very good model for respiratory diseases, asthma, chronic obstructive pulmonary disease, pneumonia. Even for surgical procedures, as I've said, laser surgeries, laparoscopy, endoscopy, and the development of biomedical products or devices, such as pacemakers, artificial heart. And it's also a very good model for wound healing, wound dressing, ointments. And just to show you, this is the biggest global animal suppliers. They are the top right now: Jackson Labs, Charles River, Taconic, AML, and China now has a big one, the Covance Research Products. And in the Asia Pacific region, so you can order your animals from Riken, from the Imamichi Institute, the Japan Shimizu Laboratories Supplies. In Australia, it's the Animal Bioresource Center, Nonam, Thailand. But most of our animals that I've shown you that we're using as animal models, we buy it from Biolasco, Taiwan. It's cheaper because of the air transport. And the local distributors, I'm not advertising, but if you're interested in purchasing, so Cygate is now the distributor of laboratory animals from Biolasco, Taiwan, and ITS is the the distributor of laboratory animals from Taconic, USA. And you can also check the IMSR. So this is a website wherein you can check the database for the different mice and also the different cell lines. So you have a vast choice in terms of the choices of the animal models. So with that, let me end my talk by stating that using the right animal model or models would translate to good science, and that would include biomedical research. So thank you.
Thank you, Doc Amy, for that very informative talk. So we now have, I guess, time for 2 questions before we have our break. So there's a question here, and there are 2 questions here. First is from Dr. Reggie De La Cruz. Can mouse be used for skin irritation and dermal irritation tests?
Actually, ma'am, the best to use is either the guinea pig or the rabbit because their skin are very sensitive. the skin of the mouse is not so sensitive. So I think if you're also planning to develop those products right now based on FDA ruling and animal modeling, you should use the rabbit or the guinea pig, not the mouse model.
Okay, another question. Are all those type of rats you mentioned as recommended, for instance, in type 2 diabetes mellitus or obesity studies already in its disease state? If yes, how about if we would want to introduce our own T2D or obese model? Can we also obtain the healthy versions of these rats?
Okay. So yes, if you buy the transgenic, this is the advantage of purchasing or using the transgenic models. So when you purchase them, you can actually do so by stating the age. In our case, let me share with you that we buy around, for the rats, we buy at around 6 weeks of age. Yes, you can demand even the weight. So there is the weight difference should be for the rats between just 5 to 10 grams. We can demand that, as well as the age. So this is the good thing about commercial vendors. You can dictate that because you're paying premium, no? And usually the rats, they develop sexual maturation at around 8 weeks of age. Yes, if you buy this knockout of transgenic models, spontaneously they develop the disease. So which means that right away, usually when we use these models at 8 weeks of age, we do 2 weeks of quarantine that I'll be discussing on animal care and management for purchase abroad. So at 10 weeks, we're ready now to use these animals say for our research. So especially on testing our test drugs or compounds. So it gives saves you time, in all honesty. And nobody would question the model because this model has been validated and you can read a lot in terms of their publication. Now, if you want to induce, which is cheaper, wherein we induce diabetes and we induce also hyperlipidemia and obesity, it takes time. So just to give you an idea for the diabetes, if you want to induce with aloxan, as I've said, you can only induce the type 1, but not the type 2. Okay? And remember, in terms of human diabetes, type 2 is more common compared with type 1. So if you're targeting diabetes modeling and you plan or intend to test your products or compounds, I think it's more significant if you test it in a type 2 diabetic model. So yes, you can induce. With aloxan, you need about 2 to 3 weeks. And the problem with aloxan, the induction is about 60 to 70%. That's my experience. So we don't induce 100% all the animals to be diabetic. So 60 to 70%. So again, it means that you'll be using more animals to induce the appropriate number. And then it takes time, the development of the disease. So like for example, for those, I've seen papers and reviewed papers wherein they are doing cardiac abnormalities. But right after aloxan injection, they checked it. No, it takes time. The development of the pathology takes time. So you need to wait several weeks after before you can use it. So it means in terms of time and resources, I think you need to do the math, which is more advantageous for you. Now, in terms of the obesity, yes, we use the special diet, a high-fat diet to induce it. My experience using the commercial high-fat diet, it takes about 2 to 3 weeks to induce the hyperlipidemia and the fatty liver that we want prior to testing of the compounds or the products. So I think it's up to you which one you would want to use.
Sige, siguro we'll have one more question. Okay lang po, Doc Amy?
Yes, of course.
So, the last question, tapos yung iba pong questions, siguro sasagutin na lang po ni Doc Amy after. Okay, so thank you. However, may I ask, is it possible to house an immunodeficient mice in an animal facility with basic equipment? If yes, how can I possibly better manage such animal using BNDG B2 MKO mice model?
Okay. I think, ma'am, I'll answer that with my next presentation. This is shorter, no? Pasensya na, the models are really more complicated to discuss. I would address that because the transgenic animals, especially for cancer, would require a more laborious or delicate management. And that would include even your caging system. So I think if you have the conventional open cages, it would be detrimental for your animals because they are immunodeficient. So if you plan to use these immunodeficient models that you have stated, you have to have individually ventilated cages, the IVCs.
Okay, so actually we still have more questions in the Q&A panel. Doc Amy will answer na lang po doon sa Q&A panel natin. So
Yes,
we'll have
oo.
again, thank you, Dr. Amy, for that very informative talk. So we'll have a 5-minute break and then we'll continue with the second part of today's session. So thank you.